This video is going to go over the histology of the epididymis, seminiferous tubules, seminal vesicles, and vas deferens. If time, I will include sperm, the penis, and the prostate at the end. However, I don't have very many pictures of those slides, so that is why they are not in here. We'll start with the epididymis. This is the epididymis under scanning power. And what you can see is there's lots of these coiled tubes. It's actually apparently one long tube. I read online it could be up to 6 meters long. And the little tiny sperm are in the center here. And you could always tell it's the epididymis because there's this border here. They don't seem to like to hang out around the edges, and I'll explain why on the high power. So we have this pseudocolumn stratified epithelium. And on the outside of it is smooth muscle, because that's going to propel the sperm through with peristaltic contractions. Then we have connective tissue on the outside. So here's the epididymis under a higher magnification. You could sort of see there's some sort of columnar epithelial. It is pseudostratified columnar epithelial. And the sperm are hiding from it. And then there's going to be a layer of smooth muscle around it. So here is high power. This is probably the power you want to draw on it. Here's the sperm, and we could see these little tiny hair-like things around the outside. And they used to think they were cilia, so they called them stereocilia. In reality, these are microvilli that are expanding the surface area, and they're nourishing and maturing the little sperm in here. Here's the pseudostratified columnar epithelium around here. And there's going to be some basal cells around the outside. And we can't really differentiate on this view the basal cells versus the pseudostratified cells. Those are probably basal cells right there. And those are thought to replace the pseudostratified columnar cells. So if we zoom in even more, this is not the picture on the page. Here is our pseudostratified cells. These would be the basilar cells out here. And we could clearly see the stereocilia. So on the page, I have this zoomed out version. And I have 6 slides here. This is scanning. We have 2 scannings. 2 low power, and then 2 high power. So this one you could probably see those basilar cells on. So go through them and draw one of them and label these structures. Our next slide is the seminiferous tubules. And like the epididymis, they are tightly coiled. And this is where the sperm is going to be made. So I've zoomed in here, and we can see here's one of those tubules right here. Here's another one of those tubules, and it has a boundary made by these supporting cells. So these cells that are forming the sperm-blood barrier are called Steroli cells, and they protect the haploid cells in here from being attacked by the white blood cells, and they do also help maturation as well. So here's a stain, not as much color, so we have the Steroli cells there and there. And then all of these cells are ones going under meiosis. We're going to have stem cells in this outer layer called spermatogonium. And these are spermatid in the center here. They are not mature sperm yet. Now we have these cells right here and right here. They're between the tubules. Those are called interstitial cells, and they make testosterone. So here we're blown up. These cells right here are the Steroli cells. And if I scroll down a little, these are the interstitial cells that are making testosterone. So these up here are going to be spermatogonium. They're going to be starting meiosis. That's becoming haploid cells. And they're working their way up. And as you can see, the cells are getting smaller and smaller. So see how much smaller those cells are. Until we have these spermatids in the center here. And again, they're not mature sperm yet. They have to go to the epididymis to mature. So again, I have several powers here. Scanning, low, and you probably want to again draw it on high because we could see the interstitial cells clearly. We could see the seminiferous tubules clearly, and that's what I want you guys to focus on. So the seminal vesicles are a gland that are located on the posterior surface of the male urinary bladder. And like all the other glands, they're basically one long tube that wraps around itself. I always think the histology looks pretty. It sort of looks like a stained glass window. And here's medium power. And we could see that it has pseudostratified cells. And then there's a lamina propria in there between the two. And then the outside of it is smooth muscle. That's going to have peristaltic contractions that propel the secretions out. So here's the seminal vesicles under high power. As you can see, there's a duct in the center. So here's scanning. It's a very beautiful looking gland right here. And you can see it's sort of like one big tube. And you can see the smooth muscle on the outside. You can see the secretions in here. So if you're lucky enough, you can see the lamina propria. You can see the smooth muscle outside and it's pseudostratified epithelium. So again, 6 views, 2 scanning, 2 low, 2 high. The vas deferens is a tube that actually does not coil around itself. How exciting. And what we're going to see is we have a mucosa made out of pseudostratified columnar epithelium. Then we have a muscularis, and this muscularis is interesting. We have a longitudinal muscularis, a circular, then another bonus longitudinal. Then you have an adventita on the outside. So if you look on the scanning, I couldn't get to the adventita even. So I again suggest zooming in. And that's a much clearer pseudostratified. Under low power, under high power, you can see even better pseudostratified. And we can see some of the lamina propria right there. So those are the 4 I want you to look at and draw. So the prostate gland is not a tube. How exciting. It's the only one that's not a tube. And it's actually these ball-like glands called tuboalveolar glands. And they make secretions that are going to be contributed to semen. There is smooth muscle between them that is going to help take the secretions from the glands into the duct, and the duct exits into the prostate urethra. So this is what the prostate looks like under histology. So here's another view. Here's the tuboalveolar glands. Here's the pseudostratified columnar epithelial lining the ducts. And I don't have very many pictures of this gland, so this is why it is not on my webpage. Our last slide is sperm. So here's some sperm. And for the sperm, we are going to have the head. We have the mid-piece, which has the mitochondria, and the tail. It's a little short tail for that guy. So here, head, mid-piece. And you usually can't see the acrosome. And what's interesting is sometimes you see a little 2-headed sperm that's mutated. This guy, I don't think the tail's on wrong. I think it's just bent, but sometimes the tail is put on the wrong place. So it's sometimes interesting to see the sperm